Welcome everyone to today's Tuesday Times Roundtable. I'm going to go ahead and introduce the session and our moderator while the rest of the crowd finishes signing in and grabbing lunch. My standard speech, just in case anyone happens to be here for the first time, we are here every single Tuesday with lunch and interesting conversation. So you're free to grab the schedule to see what we're talking about, or just blindly show up every Tuesday and trust us that you'll be here for something good. I'm Eric from the Global Learning Office, and I'm going to introduce today's moderator. Dr. Alok Deoraj is a graduate faculty and instructor in the Department of Environmental and Occupational Health. If you're an undergraduate student and don't know what that is, it's a graduate program in the Robert Stemples College of Public Health and Social Work. So if you leave here today finding that you're really interested in this idea of the health as it relates to the world around us, you may want to be interested in meeting Dr. Deoraj or learning more about that program for graduate school. And there's a very cool new building, AHG5, if you haven't been to it yet, that the Stemples School is part of. At FIU, Dr. Deoraj advises graduate students and teaches global core curriculum and advanced level courses in the area of global public and environmental health, including a very cool global learning course called Health Without Borders. His research interest is in the area of environmental health effects on the neuroendocrine control of reproductive impairment, development of cancer and reproductive origin with a focus on understanding the mechanisms of gene X environment interactions. I don't know what all that means, but some of it sounds related to what we're talking about today with the effect of the environment on reproduction. Prior to joining FIU, he worked in the biotech industry and held senior scientific and management positions to lead research teams working on the preclinical safety and toxicity screenings of anti-cancer drugs and environmental pollutants. Academically, he has more than 45 articles in peer-reviewed journals, books, and magazine and conference proceedings, and serves as an editor for the PLO's One Academic Journal. He has a certificate of management from Belmont University in Nashville, his PhD from the National Institute of Immunology in India, and a master's degree from Patna University in India. I'm very happy to turn it over to today's Tuesday Times Roundtable moderator, Dr. Alok Deoraj.
Hello, everyone. Good afternoon. And I'm really pleased to be here. I think this is my third Tuesday Times Roundtable, and I always enjoy to be here and talk to the people and moderate something very burning or very relevant topics which pertains to environmental health, because I am from Environmental and Occupational Health, and as he just mentioned, at the Robert Stemples College of Public Health and Social Work. And we offer a master's in public health as well as PhD in the public health discipline. And why? Because see, as you just mentioned, this is a beautiful building, just opened. I'm really proud of it. And here we have Mike Reed, who has been the project manager, who has been very, very involved in this building. So if you haven't been to this building, please come and see and visit us. So we would be really glad to welcome you there. And even maybe Mike or somebody else can give you a round of that building. So it's this really, really cool building. You must come and visit. So we do offer a public health degree into master's as well as the PhD. And it is very interesting because we are at the time when, if you look around everywhere, it's the prevention what we are talking about. And public health is primarily for the prevention of any kind of a diseases or any kind of a hazard experience or hazardous exposure you have, how to prevent that. And that's a very upcoming area, and it had been there for quite some time. But this is very cool if you can, if you are interested, those especially who are senior and don't know where they want to go, this is a good spot to explore. And right outside, by the food court area, we also have a booth from our department. If you want any information about our program, how to apply, what are the requirements and everything, you can grab that information from there. And this is another important thing. Whether people call me biased or whatever it is, because I do feel very strongly that environmental and occupational health is the core of the public health profession. Because this is the discipline which tells you that how things happen. And until and unless you know how things happen, you cannot develop any prevention or mitigation plan. So If anyone who is interested, please come get information and explore this opportunity for you. Coming off to the environmental and occupational health, now many of you already have this title for today's talk. It's just a beginning because we are very, very concerned about the pollutants and the environmental pollutants and toxic substances which is around us. And this article which in New York Times, which appeared very recently, and it caught my eye, and I think that, that sounds like a pretty good idea to have some sort of a moderation on this burning topic. And as you can see, this is just about the story of bisphenol A, which is nothing but it's a kind of a plasticizer. And it had been in the news for quite some time. And scientists and as well as people are not sure how toxic it is, what it does. And that's the challenge we will also be talking about today. And I'm not going to be giving too much of information. Rather, I want you to ask questions and have some sort of a discussion about how or what kind of a concern you have about the chemicals in your environment. But just to summarize, this is the story of bisphenol A in this paper, what you have. And as you can see here, that's the actual paper based on this New York Times article was written. So Jodie Flaws, actually, I have met her. She is at the moment, she is in Urbana, Illinois. Her work had been that she injected this bisphenol A, which is an environmental pollutant, and it is used heavily into the plastic industry. And what she found was that it damages the developing ovaries. It damages the developing ovaries, and she injected it the dose comparable to the environmental exposure people are having in the environment at the moment. So based on that, her findings in mice was that could damage the developing ovary. What she also found was the follicles in the ovary of treated mice were fewer and smaller. And she found that in another study, BPA treatment stopped viable egg production in mice at a young age. Then childhood exposure may contribute to female infertility, and that adult exposure may result in a shorter reproductive lifespan. And FDA has banned this, its use on baby products here in the United States. But it is still being used in many other products, we are using on a regular basis. Even if you think from the very morning when you get up, and how many chemicals do you come in contact with from the very beginning, like right from the time when you start to brush your teeth or wash your hand, detergents or anything of that sort. Then you apply the cosmetics, the nail polish or anything, whatever you get exposed to. On an average, you get exposed to about 3,000 to 4,000 chemicals on a daily basis. Besides the things what you eat in your food, what you have in your plate, the air you breathe, the food you eat, the water you drink. So there are so many chemicals around us. And we don't know what it is doing to our body and what is happening. What are the consequences? Is there immediate consequences or the long-term consequence? How long it will take to show any effect? What stage of the life you are, whether you are a young child or you are still an unborn child. You are a pregnant mother or adult or a senior or you're an elderly person. Where you are. And depending on that, how you're taking those chemicals, how you're getting exposed to, and then how it is impacting your health in the long run and the immediate term. So this is just an example, and there are so many of them. We'll talk a little bit in this one. This is just to prove the point that, okay, mice, we can consider that this is a little bit away from the evolutionary tree. So this study was done by Patricia Hunter in Washington State University, and she did the similar studies in monkey, in rhesus monkey. And what she found, there also exposure to BPA in the second and third trimesters of pregnancy of rhesus monkey altered follicles and oocytes, the germ cells that develop into eggs. It is highly likely that similar effects may occur in developing human females as well. And it is no secret that over the period of time, all over globally, it is being found that the exposure of chemicals is linked. I mean, it's still, it's not biologically fully proven, but it seems to be linked to the low sperm counts, the precocious puberty, or the delayed puberty, many types of cancers, the mutagens, and all those kind of things. It is linked, and that's the challenge we have, because there's so many chemicals around us, and no toxicity data available. And that's the challenge we are at the moment, and how we are moving into the 21st century. I'll be addressing into this talk today. So, this is the 5 questions we'll discuss today. And what are those? What kind of toxic pollutants are there in our environment? When these pollutants are of more concern, what do we know about them? How do we know about their hazardous effect on human and ecological health? Who is screening the toxic effects of environmental pollutants? So these are the 5 questions we will be discussing today. So first of all, what kind of toxic pollutants are there in our environment? There are so many of them. There are some natural ones which are there. There are not only that, there are chemicals, even the traditional ones, the greenhouse gases, the ultraviolet radiations. There's so many toxicants, the hazardous things which are there in the environment. The most concerning is, and we'll talk about that, but here I'm just trying to highlight that, look at this, whether it's the baby toys or the smoke or the lipstick or the radiation or in the cans or the battery, the lead, and even the toxic waste is dumped into drums and all that. All the dumping yard where we have all these toxic waste which has been dumped. The corrosives, the acid, so all these kind of things are there in the environment. It all depends where you are, which part of the country you are, which country you are, which part of the world you are, which part of the city you are, how you are getting exposed to, how long you drive, what kind of breathe or air you breathe. So all these things, like here is just kind of an example. So, and here it shows the different stages of life. Each one of us get exposed in a different amount or in a different quantity and those kind of things, and it all depends on the stages of your life. If the baby is in the womb of a mother, or the infant who is playing all the time with the toys which are made of plastics, the teether, or anything like that. So we are getting exposed at a different amount in a different stages of life. And certainly, most certainly, the children are the most vulnerable ones. Just you have to think about in this term that the surface area, the same amount of pollutant is there in the environment. You have a bigger surface area to absorb that, but the child is small. And then they are getting the same amount of exposure, the same amount of pollutant they are getting exposed to. Now, they are at the developing stage. The maximum development or the faster rate of development, which is there in the first year of the life. And if the baby is exposed at that time, this has long-term consequences. The baby in the womb, if get exposed to cocaine or tobacco or any kind of things, that has a long-term consequences, possibly starting with the low birth weight, but maybe it may not manifest it right away. The manifestation you will see 30 years down the road, 40 years down the road, because that's when the genes which are there, they get programmed. That when they need to, if in the normal time, this is the time when the precocious, when the puberty has to trigger in. This is the time when the nails have come in, or the eyes have to develop, or the brain neurocells have to develop. But these environmental chemicals can alter those temporal and spatial expression of those genes. So that may or alter the normal development, that can alter the development. So the imprint or the things what can happen in the womb or at the earliest stages, the effect what you may see down the road, 20 years down the road, 30 years down, and that is also one of the things we will be touching on that as well. What happens is that those small amount what you intake, not necessarily they will show you the effect right away. It gets accumulated over the period of life. And that is why all the complex diseases such as cancer, cardiovascular disease, diabetes, generally they happen towards the later part of the life. So what happens? So let's assume that the environment is polluted, or it is polluted in many ways. And what kind of pollutants do we have? It could be physical, it could be biological, or it could be chemical. But there are only 3 ways you can get exposed to, and those are either you can inhale it, something you inhale it, or you ingest it, or touch it. The ingestion also comes like water or something, what you are drinking. What kind of a pollutant is there? Where the water is coming from, whether it's clean or not, whether there was a groundwater contamination from fracking, or it's the oil industry which has dumped a lot of chemicals. Was there a leak into the chemical dump which has infested the river stream from where the water is being pulled and it is not being treated? So what kind of a water you are drinking? What kind of a food you are eating? What kind of a pesticides have been sprinkled on that to keep it fresh? The shelf life is increased so that it can be transported from California to Kentucky to Maryland, to Europe, from Europe to United States, so that it can stay longer. So insecticide, pesticides, all those kind of things which is there in our environment. But these are the three ways only you can get exposed to, either inhalation, ingestion, or by touch, when you touch it. But it all depends on the magnitude, frequency, and duration. How much you are getting exposed to. How much you are getting exposed to, how often you are getting exposed to, or how long you are getting exposed to. So our ecosystem, as well as our body, is a very, very robust system. Until and unless you abuse it, it is not going to give in. It is very, very robust system. But if you abuse it in terms of that you are binging or you are taking something on a regular basis, it can also relate to your occupational hazard. Like, if you're getting exposed to, you are working in a chemical industry and getting exposed on a regular basis, that will have a health consequences sooner compared to the other people. If you're working in a coal mine, if you're working in a chemical industry which is producing toluene or benzene or something like that, you're inhaling it all the time. That is why one of the reasons why people took out the lead from our fuel now. We have unleaded fuel. Because the people essentially, earlier it was the people who are working onto those gas stations, they were getting those cancers. So all those kind of things. So there is a hope, or there is also how you can minimize the exposure going forward so that those health consequences can be mitigated or can be eliminated. Eliminated, I wouldn't say, because risk cannot be eliminated from your life. It can be only minimized. So the magnitude, frequency, and duration, these are the three things what you have to keep in mind. And these are, again, either you can inhale it, or you can ingest it, or you can touch it. So the left side is the lung system, the middle is the ingestion thing, the digestive system, alimentary canal, and this is the touch, and the dermal system. So this is where the first introduction to any chemical or compound, when it comes to what it is, it will be your cell. It will be at the very, very cellular level. That's when the touch or the ingestion, that's when it will impact. How many cells get impacted, how often they get impacted, whether it's going to cause the DNA damage, whether it's going to affect the cellular machinery, whether it's going to cause the cellular growth, uncontrolled cellular growth to cause cancer, or it will disrupt the programmed cell death, or it will even trigger some of those genes which are not supposed to be expressing at that particular time. So this is where, whether it's by touch or ingestion, the moment those chemicals go, this is the process that starts. The cellular machinery is triggered. If it is mimicking some of our own molecules, such as estrogen, that creates another problem. And we will talk about that as well. BPA is one of them. And here, just an example, the farm workers who get on a continuous basis, if they get exposed for a longer period of time, how much if they, look at this person doesn't have even the protective mask or anything. If he is continuously, if this is his job, if he is getting exposed to this pesticide on a regular basis, this will have impact on his life, his family's life. He goes, if he doesn't wash his hand properly, he touches his child. He embraces his wife. And that's how these chemicals can get transmitted from one person to the other person. On a regular basis, that's what you have to keep in mind. So that's what I was talking about. One was the magnitude, frequency, and duration. And the impact what you see if you have an acute exposure. And what is acute exposure is, acute toxicity refers to a chemical's ability to cause harm as a result of one-time exposure to a relatively large amount. Anything can be a poison. Anything can be toxic. If I ask you to drink 2 liters of Coke in 5 minutes, if you are drinking, like, on a regular basis, it's not going to hurt you. But if I ask you to drink 2 liters of Coke in 5 minutes, or if I ask you even to have sugar, 5 or 2 pounds of sugar in 10 minutes, that will be the acute exposure. That will certainly imbalance your whole physiological system. So that is the acute exposure. But then you have chronic toxicity, refers to a chemical's ability to impair health when repeated low-dose exposure occurs over a long time period. And that's what, in many cases, is happening to our world. And what happens is that even if you have a chronic exposure, and that's what we will be going there, we will talk about this one, that over the period of time, if the body is not able to clear it, there are 5 steps which happens. Any chemical, what you get exposed to, it gets absorbed, then it gets distributed into your body, A, D, then it gets metabolized, M, E, metabolized, and then after that 4 steps actually, and then it gets eliminated or excreted out. But these chemicals, and I will be going there, you will see most of those the chemicals which has originated from the anthropogenic activity, man-made activity, those are the hardy ones. Those are persistent. They bioaccumulate. And then not only that, they biomagnify. And the impact is even much more. So those are synthetic chemicals, starting with the DDT. Those are the synthetic chemicals, pesticides or insecticide, or for example, many of those plasticizers, the BPA, PCBs and all. So those are very, very persistent. They don't go away. I'm not saying that everything is bad about that, because that's what have also improved our quality of life. We are comfortable sitting here, nice chairs, the hardy wall or your wood, but you are also getting exposed to. So you have to find a way whether what is toxic or not and how it is toxic. So it has to come, so the toxicant or the pollutant is there in the environment. So the environment is the one component, the another is the toxic agent, and you have the person, what age, where it is, even the organism, whether it's a plant or animal, how you are interacting with that. So all of this have to come together in order to manifest any kind of a hazard of health to you. So what we are afraid of, pollutants. So why we are afraid of these chemicals? Because the pollutants, toxicants have the ability to cause harm to environment, ecosystem, and our organs or biochemical processes in our body to cause health hazard. And that's why we are afraid of. But the problem is that many of the chemicals, we don't know what actually it is doing. We know that it has increased the shelf life. We know that it has made the tomatoes much more hardy. We know that these wooden thing has become very, very strong. We know that this plastic is much more pliable. Now we can have probably the foldable cell phones. But we don't know exactly that how it is going to have or what toxic effect it may have. And that's the challenge we all are facing at the moment. And this is just one of an example. Do you know that we have in our environment close to about 100,000 chemicals, synthetic chemicals I'm talking about. Close to about 100,000 chemicals. And we know for only 3,000 to 4,000 of chemicals, the toxic effects of only 3,000 to 4,000 of chemicals. The rest we don't know. And on an annual basis, we are dumping in our environment 1,000 to 2,000 chemicals on a day, on a regular basis per year. The reason is that we find some chemical, we find something which is going to improve our lifestyle, improve our, like maybe something even which prevents the spread of some germs or something. But that is a chemical. We don't know. We don't know that whether it's going to affect, whether it's a carcinogen, whether it's a mutagen, whether it's an endocrine disruptor. We don't know those things. And that's also going to be the topic of my talk today. And as you can see here, toxic chemicals in your home, PFCs, BPA, formaldehyde, phthalates, toluene, PBDE. Toxic chemicals in our body, BPA is found in 9 out of 10 Americans. PFC, PBDE, and phthalates, even the BPA has been found, or PCBs have been found in the breast milks. even though the people have not been that exposed, because these have been in the environment. There's some natural source as well, but these have been in the environment because they're so persistent. They don't go away. So that's the chemical. These are the things what you can see here. They are putting your health at risk. The fertility problems, asthma in the long term. It is also affecting Parkinson's, Alzheimer's, schizophrenia. Then we have 232 toxic chemicals were found in umbilical cord blood from US neonatal. So you would think that the babies may not be exposed. They are. So they have been found into the umbilical cord blood. And here, rise of synthetic chemicals. It all started after World War II, the industrial revolutions when it took place. Because that's when all the synthetic chemicals started to come into the environment. And that was widespread synthetic chemical production after World War II. People are largely unaware of the health risk of many toxicants. And potent insecticide DDT was spread widely in public areas, even to prevent malaria. And that is when I think many of you may have heard about the Rachel Carson. She was one of the pioneers into the environmental movement in the 1960s. So that was, she wrote also a book, very famous book. It's called Silent Spring. So this is again DDT used by the military. North Korean prisoners of war were sprayed with DDT powder by American soldiers. They have been banned now in US, but still it is found in the human blood and urine and different things. So that is about the DDT. The plastics. How comfortable it has made our life. Everything disposable, the styrofoam, the Ziploc bag. And I mean, I use it too. I mean, it's so convenient, right? So, but the plastic, 1907, Leo Baekeland developed a bakelite, a form of thermoplastic. World War II, US military used bakelite to produce lightweight weapons and machinery. Saran wrap was a World War II invention, which is so convenient. It does have some advantages. It protects the food. It doesn't let it spoil. This is again throwaway living, post-war plastics. So everything disposable. But what does the plastic have in there? And as you can see, additive like nonylphenols, PBDEs, phthalates, and bisphenol A are added to plastic during production of catalyzed monomers into polymers, give it different properties like flexibility and durability. And color, these chemicals are released from plastics and enter the environment. Additive even contaminate the foods they are designed to protect. So those are the additives which goes into the plastic. And that when they leach out in your food, in your water. And that is why you must have seen that everywhere. In fact, in the flyer which went around, it shows the BPA-free things. The people are trying to say that, okay, there is no BPA there. But so you can have a ratio more or less, or you can reduce the ratio of those plasticizers, but there has to be something. There has to be some synthetic chemicals in there. This is just a landfill which is filled with all kinds of plastic waste. And it is very difficult. In fact, I was telling somebody, I read an article or somewhere about that it doesn't go away. There was one plastic bag which was hung onto a tree by a building, and that stayed there for almost 10 years. And nothing happened to that. And the lady who used to see that, she created a Facebook site for that. And she monitored it for 10 years. And finally, one day it had gone, but not because it got degraded, it just may have flew away. But she celebrated just to make a statement that it stays there, it doesn't go. If you Google it, you will find that. So, what are those chemicals, those man-made chemicals which we are more concerned about? And those are carcinogens, mutagens, teratogens, allergens, neurotoxins, and endocrine disruptors. These are the broad category. There could be other categories also, like corrosive or acid or oxidative stress causing and all those kind of things. But carcinogens, mutagens, teratogens, allergens, neurotoxins, and endocrine disruptors. So those are the chemicals, the categories, the broad categories what we have. And this is the environmental agents produce mother to fetus epigenetic effect. And those primarily are either the teratogens or the endocrine disruptor. Endocrine disruptors, dioxins, incineration, which comes out because of the dioxin incineration, pesticides, bisphenol, phthalates, synthetic estrogens, diethylstilbestrol, phytoestrogens such as genistein and quercetin. So those, what are the endocrine disruptors? How many of you have heard about the estrogen? Raise your hand. You know that, right? So it's a female hormone. But these synthetic chemicals, what happens is they mimic the estrogen molecule. And when they mimic this estrogen molecule, they bind to the same molecule what is supposed to be binding to the natural estrogen. So now think about it, that when there is no need of this estrogen to be binding to trigger some cellular response, it is binding to that molecule when it is not needed. So the cellular machinery starts without even its requirement. And that is what many times the estrogen has been, not many times, it has been proven that that is also the cause of many, whether environmental estrogen or even your body estrogen as well, because your body burden of the estrogen over the period of time, it increases in female. So what happens? And that's what is the source of the breast cancer or ovarian cancer or endometrial cancer and those kind. So now you add to that all these kind of things what is there in the environment. That is a problem. Not only that, even for the males, androgens, there are some chemicals which mimics those. So that causes problem. Then if the baby is in the womb, if that is getting exposed to, and that is why if you go and find the studies, you will find a lot of places we have the precocious puberty. United States and many parts of the world as well. But at the same time, you also have a problem of the hypogonadism. So those kind of things are there too. So examples of some of the hazardous chemicals are here. Air pollution. Air pollution is also very, very prevalent in all different parts of the world. China, India, sub-Saharan region, and all those places where indoor pollution is much more higher. Even here also, I would say that people have a lot of indoor pollution, but there, especially like, you know, the people who are cooking the food inside. So all the bisphenol A and PCBs from those forest fire or the wood chips or the coal, what is being burned inside the house, that is carbon monoxide and all those kind of things. The air pollution is causing the problem, indoor pollution. Not only that, the developing economies where we have now much more cars and all that, and on a regular basis we have advisory that please remain inside because the air is so thick with all this sulfur dioxide and ozone and all those kind of things. So air pollution is a problem there. So this list also include some of those things which the World Health Organization has issued for all over the world, like, you know, which is causing problems. Then arsenic, asbestos, which causes asthma and all that, and even also it is an occupational hazard as well. Benzene, BPA, cadmium, DDT, dioxins and dioxin-like substances, highly hazardous pesticides, because all the farm workers, or even for us, when we buy our stuff, they are not properly washed. If you don't wash it properly, still there will be something remaining there. Inadequate or excess fluoride, lead, methyl mercury, PCB and TCDD. So these are just some of the examples. So as I just mentioned, the approximately 100,000 chemicals of anthropogenic origin are of major concern. So those which are there in the environment, slowly or gradually it may degrade. But what humans have created, that may stay here for a long, long time. The reason also for that is that the bacteria or the biological system who is able to degrade has evolved along with those pollutants which is being created or which are there in the nature. But man-made chemicals which are very recent, so the bacteria and those kind of a biological system has not evolved or has not been able to degrade those from the environment. So that is one of the reasons why they just stay there. They're persistent. And they bioaccumulate over the period of time because they're not getting degraded, right? So we don't know. The biological and toxic endpoints are not known for those chemicals. Their persistence, bioaccumulation, and biomagnification in our body and environment is not known also very well. Then the goal is it is a daunting task because we have so many chemicals already in the environment, and we are also creating or making those synthetic. Don't just think that synthetic chemistry or synthetic chemicals are only for the engineering part and all that. Think about those pharmaceuticals, what you, on a regular basis, the drugs and all that, what you are having to treat a disease. But that is also going into the drain. Do you know that the medicine, what you take, most of it just goes into your urine and just passes out? Because not all of them are being used. So that is also causing a problem. So, and those are, most of them are man-made or the human-made. So that is also there in the environment. Even the contraceptive estrogens which are being used, which goes into the environment, that causes problem. Even here in Opaka or Okapa, what is the place in Florida? It's called Opaka, right? Opaka Lake or somewhere. So that's where there was a chemical spill, and the estrogen-like chemical which went into the lake, and after 3-4 years, the alligators, majority of them turned into females. Not only that, even they had the small part of the testis, but they turn into females. But the beauty is that if you take remedial measure, and that's what I was telling, that our ecosystem and our body is a robust system. If you take a remedial measure, it will improve, it will get better. And that is the hope. So, and as you can see here, this is where, like starting from organelle, biomolecule, then a cell stage, the organism stage, then you have population, community, ecosystem, and biosphere. Why I am showing this thing is, and as I just mentioned, that the first response to any chemical or anything what you get exposed to, it starts at the very, very molecular level, at the cellular level. So there, if you have the knowledge, traceability, ability to assign caution, proactivity in use of knowledge is high when we start at the cellular and molecular level. You start to correct, you first find out what is the toxic effect at the cellular level. But the traceability is low when you are at the higher level, long-term consequences. And this can take even thousands of years to show the effect, but it starts from the individual cell and it just goes all the way to this is the organization of this whole ecosystem, as you can see here. Ecological relevance, response, duration, long-term consequence. And this is the thing which is from the Landis and Yu 1999 paper. And as you can see here, that on the x-axis, what you have is the spatial scale. And on the y-axis, you have the time point. So the response, or what you get at the 1 micrometer distance, is in seconds. Like if something is hot, you immediately come to know. If something is burning, if something is itching, you come to know right away. But as it goes into the 1 millimeter genetics, molecular organ and tissue, it's about 1 meter. Like you would not start to feel immediately sick. Maybe once you have a restaurant food, the shrimp toxin has gotten you ill, but it didn't happen immediately. When you came out of the restaurant, probably even on the Facebook, you posted that, oh my goodness, that was an excellent restaurant. But when you got home, what happened? You had a loose motion or you start to vomit. So it takes time. It may take 1 hour, 2 hours, 3 hours. But then maybe, possibly because of the same bug, the other person also gets it, even though the person didn't visit that restaurant. So that maybe even take 1 day, 10 days, something like that. Slowly, if it is not prevented, slowly, if it is not checked, it can even spread into the population. And the perfect example at the moment is the Ebola, what is going on at the moment. I will not go in there, but I'm sure that there may be a lot of questions there. So as you can see here, organ and tissues, but the time, as you see, landscape, ecosystem dynamics. So even until the 10,000 kilometers square, do you know that the sub-Saharan dust storm affects the Florida weather? But it takes time. It doesn't come right away. So if some pollutant is the other part of the world, can also come your way as well. It may take time. And that is why the climate change has also become an important issue, because you don't see it right away. It may take time to show its effect. But first response is your cell. First response is your molecule. First response is your cells at the individual level. But the challenge what all of us are facing at the moment is how do we know with all the so many chemicals, how do we know the toxic effects of those chemicals? And what is happening is that's the biggest challenge for our pharmaceutical industry, for our regulatory bodies and all that. We cannot go and injecting human beings to find the answers. So what has happened is the regulatory agencies or the pharmaceutical industry, what they're trying to do is that they are using all kinds of biological system, whether it's yeast, worms, flies, fish, mouse, primates, all of them, to get some information at the very, very smaller genetic, molecular, cellular level, and then trying to extrapolate the information to build a story that that may be happening. And that was one of the things what Jody Flaws has mentioned, or even the article what was posted earlier. Earlier, the studies were much more into that it has to be done only in the mammalian species. It has to be done only into the rhesus. Then only people will understand and people will say that directly relate to the human beings. But in the last 10 years or so, the paradigm has shifted. The toxicity trial or testing is on this premise at the moment is that if it is harming even a cell, if it is harming a bacteria, if it is harming a protozoan, or if it is harming a yeast or zebrafish, it is also has a toxic effect in the long run to the human beings as well. So you have a consortium of these models from where you get this information and try to put it together and make sense out of it. And that was one of the things what I was also doing when I was in my previous position, that there we were trying the preclinical trials for the toxicity of the anti-cancer drugs. Whether what it does to the developmental profile, what it does to the cells, what it does to the arteries. So you monitor into different organisms. But what happens, and this is what, like if you go onto this direction, if you have a smaller molecule or flies or worms, they also have a similar mechanisms. So there you have a high throughput. You can have a multiple individuals, 300, 400 individuals to test it. But that may be away from the mammalian species. So the cost is less if you are in this direction. The cost is higher when you are trying to go into the primate system. And that's one of the tools, I would say, for the 21st century, where you can go into these smaller molecules and then analyze those biological systems, that what is happening to the human beings. Because, for example, zebrafish has 85% of homology of its genome with the human beings. There are lots of processes, the cellular processes, which are the same as human beings. So if you try to understand, if you understand what is happening at the zebrafish level, possibly you can extrapolate those information to the human beings. So modern approaches of toxicity assessment. What are we doing? So what we learned, or what we talked about so far, what kind of pollutants are there in the environment, what is of the most concern, where they are, and when they can affect you, we talked about those things. So now how do we know it? And that was one of the slides which I showed, that we can use some of the models which are even lower into the phylogeny profile. So the modern approach, now throw in the computers. And that has become a big tool even. Because we are publishing on an average maybe 6,000, 7,000 publication on a single topic on a regular basis. Those data are there. So the computer has also become a useful tool to combine this information and then attach with your model system, and to find and extrapolate the information to find out which chemicals are toxic into the cellular profile or cellular mechanism. So what it tells you, combination of in silico and in vitro assessment. In vitro assessment means that you are testing the toxicity into the petri dishes or the test tubes, not actually into the animals. So what you are doing, you are reducing the use of the animals too here. You are just testing it into the in vitro system. And in silico is that after getting the information, you are putting that into the computer to generate certain kind of models. And then say that, okay, this thing is going to be toxic. And it is needed, it is very much needed to cost effectively and in a speedy way to find the toxicity of so many chemicals which we don't know the toxicity about. So To understand the changes in biological processes, oxidative stress, epigenetic effects, inflammation, cell cycles, cell death, uncontrolled growth, interruptions in signal pathways at environmentally relevant concentrations, trigger events that leads to adverse health effects. So once you know that what is happening at the cellular level, that may be causing the health effect down the road. And these are some of the tools of the molecular biology, toxicology, genomics, and proteomics. This is what is called the microarray, where you individually, you can test what genes are involved, what is happening when you treat these cells with certain chemical, what is toxic. So certain genes will light up, certain genes will not be lighting up. And that shows that those genes are involved into causing the toxic effect or causing the health effects. And once you know that, you can build this story, and those genes, those hormones can become the target to find a diagnostics or to find the treatment. So there are different ways. You all know that human genome was sequenced in 1999, 2000. So genomic approach has become the more advanced in 21st century going forward, using those to find out the toxicity of all these chemicals. And that will be faster too. Earlier, the traditional way was that we used to use all kind of rats, dogs, cats, alligators. All those kind of things were used to monitor what is happening, and that used to take a long time to find this out. So we are sort of, in this time, we are empowered that we have these technologies to move faster. Of course, there are so many other things which comes into play. Who is where, which person gets the funding, and all those kind of things are there. But at least it allows you to have these tools available or by which you can move faster. And this is just a summary of what we can do with these new tools. And as I had just mentioned, the molecular biomarkers. So in the previous slide, what you saw here, from here, we cannot pinpoint one gene or one hormone because it's an interaction which takes you to which gives you health hazards because of the exposure of certain chemicals for a long period of time, to give you a health hazard for the cardiovascular disease or diabetes or cancer, breast cancer, colon cancer, and all that. It's an interaction of different genes which are being triggered by the exposure of these chemicals. So there you find these molecular biomarkers. And once you find these molecular biomarkers, which represents to inflammation, disrupted signal transduction, endocrine disruption, angiogenesis, and all that kind of cellular machinery, which is leading to all this integration of environmental exposure effects, data from molecular epidemiological approaches, leading to diabetes, obesity, abnormal lipid, metabolic syndrome, cardiovascular disease, and all that. And here, exposure to environmental factors. So toxic chemicals are just on the top of it. Reproductive factors, diet and nutrition, psychological factors, drugs and alcohol, all those things are affecting these cellular processes. And on the right side, that's where the computer comes in. What you see, the bioinformatics, the DNA, RNA, how they are interacting, which time, what time those genes are being expressed. So once you know, then you can find out, well, at this cellular level, this particular gene is interacting with certain genes to cause this disease here, whether it's obesity or cardiovascular disease. So this is the way or the direction the field is moving to find out, because there is a push that we do want to improve, like we do want more new drugs, we do want pharmaceuticals so that we can treat the diseases and infectious diseases and all those kind of things. But that's why you may have heard about a lot of the side effects. So people come to know, and then it gets retrieved from the atresin is there too in the drug discovery areas, once you find out. So, but even this is not the foolproof. It still has the limitations. And what are the limitations? Establishing direct health effects of pollutants due to complex exposure, including combinations, genetic makeup, diet, lifestyle, and smoking. You have to also remember that you are not getting exposed to only BPA. You are not getting exposed to only PCB. You are not getting exposed to only DDT. Probably you are getting exposed to everything, right? So what is this complex exposure doing? How the body is processing? It is also possible that when it gets metabolized in the body, the metabolites, the broken part of those chemicals, can become even much more potently toxic. Or it is also possible that the other chemical is nullifying the other one. So those are the questions we can ask today with these kind of tools what we have. Now, you have these tools, you got the DNA, gene, RNA from urine sample, blood sample, hair sample, semen sample, and all that. You were testing all these things. But you didn't know that this person was also smoking, or this person already has a pre-existing disease. So those are the covariates or the different kind of factors which also comes into play. So you can say, and that's where it throws you off. And those are the limitations are going to be always there. The population even today is so many mixed. So you cannot even say that, okay, this is for the Hispanics, this is for the Caucasian, this is for the Afro-American. So the complexity has increased. But still, we should be hopeful that we have the tools by which we can combine all these things to find some meaningful answers. And that's the challenge. And that's just I was telling you, that it is also possible that you are not getting exposed only with the food. You are also probably getting exposed something from your makeup, or you are getting exposed from vegetables, play, work, or home, somewhere. And these are the different things, what you can see here. This is the industry, which deposits on the ground, the eating vegetables, inhalation, soil ingestion, dermal absorption, eating fish. So multiple pathways can come together. And that is what the risk analysis exposure science which is going into their area. Also, these chemicals, and I had just mentioned the bioaccumulation, that planktons or the plants which are lowest into the trophic level, they have, let's say for example, 0.000003 parts per million. When it goes to the fish or the other higher trophic level, 0.04, 0.5, 2 ppm and 25 ppm as it increases into the trophic chain. What does that mean? Just like when you condense the calorie, it's the same thing, you're also accumulating those chemicals as well as you move into the trophic level. And certainly, because it is persistent, it doesn't go away. The system is not ready to get rid of it. It is not getting eliminated. Now, so this is about so far the chemicals, how we are getting exposed to, what we are doing with it, how we should know that whether it's toxic or not. So we talked about that. Now the last thing is that who is doing what, or how or who is involved in finding these things out. And we have National Toxicology Program in the United States, which is a part of the EPA. Then we have Future of Toxicity Testing Work Program, that is also EPA. National Toxicology Program is there. Agency for Toxic Substances and Disease Registry, this is from CDC. Those people are involved. And there is a public-private partnership, university and all those people, they are involved into these kind of testing and finding it out and using these tools to move quickly. Because if you don't, new chemicals will be again putting into the environment. And these websites are there. If you want, you can just go ahead and take a look. This is OECD guidelines for testing of chemicals. This is the European and other places. So in order to harmonize that the information, what is, we find it here, how it is also applicable to the other parts of the world. So this effort is also being made. So OECD is an European agency which is working on that. So these are some of those organizations which have developed certain guidelines that how, like, it's not that you can just go and dump these chemicals and see what is happening. Specifically, they're asking whether these genes are involved or not, because those pathways have been worked out. Those physiological systems have been worked out. So you just cannot say that, okay, this is going to cause the neurotoxicity. This is going to cause the angiogenesis problem. So there is a certain set of guidelines. And for the United States, NRC, National Research Council, had just 2012, they have published the whole guideline for this thing. And this is the 21st century moving forward in using this genomic data going forward so that cost effectively and in a speedier way we can find the information to what toxic is, what is toxic, what is not toxic. So why we are doing all these things? The anticipated outcomes is the cost-effective and faster screening of chemicals help prioritize efforts to pollutant chemicals of greatest risk. Because we just cannot go straight to the monkeys or rats or mice because that will be very, very expensive. But if you take this high throughput screening methods or the lower phylogeny and even the in vitro system, the cell culture system, you can prioritize, you can identify certain cellular link which may be leading to the cardiovascular or may be leading to the endocrine disruption, which are much more vital to test. So you can prioritize those chemicals. This seems to be much more potent toxicant. So let's focus on that. Let's find, then you can reduce like 10,000 chemicals you can start and you can do it, but ultimately maybe 5 you can take it to monkey. So that kind of helps you moving forward. Then possibly more scientifically relevant information, because it's just not showing the association by data logistic regression or those kind of association. This is actually looking for the biological plausibility. What is happening at the cellular level? What is happening at the DNA level? What is happening at the physiological level? So it's just not that there is a disease cluster there because there is a chance or the coincidence that those people are there and the effluent is coming from the industry and there is a disease cluster there. That is also important. Once you know, then only you can go and find that out. But if with this approach, you have much more biological plausibility. You can for certain might say that, yes, this is what is causing the problem. But certainly, as I just mentioned, the limitations are always there. Then we have influence policies for regulatory, economic, advisory, community or technological intervention. That is very important too, because that's what in public health area, environmental health and all that we do, that we just cannot go and shut down the industry. We just cannot go and shut down the production of the plasticizers because on that, there are so many livelihoods are also resting. And that is why what I'm saying is that what you have, that with these information, empowered with these information, the scientifically sound information, then only you can influence the policies for regulatory, whether once you have that information, then only you can say, oh, like 0.05 parts per million is not causing, so maybe we can reduce so that the person can emit more, or we found no, that's too much and that is causing the problems much faster. So the regulatory policy might go and adjust those dose level, and maybe you all may know that here even your water supply, the utility, it's mandated that they send you every year, they send you what is there in your water. So it's an acceptable level. But maybe tomorrow with this information, those acceptable level can be even reduced. So as the new information comes out, that can influence the policies. Then the regulations will be even in place. So regulatory, economic, advisory, community or technological intervention, technological intervention, what we need to improve the engineering side so that the air handling system gives you much more cleaner air where you live. Or you are working in a very hostile chemical environment. What kind of a HEPA filter needs to be put in so that when the air is circulated, it comes clean? So technologically, how you can improve those things. So those information can also influence that. The goal is how to ensure safety, to minimize human health risk and the environment. That is the goal, to do all these things. And the purpose is to reduce the use of hazardous chemicals. How you can achieve that? With these information, possibly you can reduce the use of hazardous chemicals such as BPA, PCBs. PCBs refine the synthesis process to make the chemical less toxic or biodegradable, replaced with non-toxic substances. That's how you can do that. I'm almost done. So that they do not persist and accumulate in the environment beyond the level that is predetermined to be safe. But this is the final slide just to give you much more hopeful that chemicals only contribute to the 2% of your health hazard problem. It's the poor diet which causes much more problem. Okay? And right in the beginning I had mentioned this is in the 16th century. What is that is not poison. All things are poison. Nothing is without poison. It is the dose only that makes a thing not a poison. The remedies can become poisonous, can become toxic. That's all for today. Any question?